Welcome back to Dr. Liu's VCE Biology. Today's topic is responding to antigens and acquiring immunity, as presented in the 2025 NHT Biology exam. Let's learn together. If teachers, parents, or students notice mistakes in my explanations, please provide me with your feedback. Correct answer. C. The setup occurs when plasma B cells produce Ig antibodies, which then bind to receptors on the membrane of mast cells. Act 1. Sensitization. Before the symptoms, first contact. An allergen, pollen, dust, enters the body for the first time. The factory, plasma B cells recognize the allergen and produce specific Ig antibodies. The priming. These Ig antibodies travel through the blood and dock onto receptors on mast cells, the loaded gun. The mast cell is now sensitized. It is coated in antibodies and packed with histamine, waiting for the allergen to return. Act 2. The reaction. Upon re-exposure, degranulation. When the allergen returns, it binds to the Ig already sitting on the mast cell. This triggers the mast cell to release histamine, causing the physical symptoms of an allergy. Why the other options fail? Option A. Histamines are not found in peanuts or dust. They are produced and stored inside your own mast cells. Also, allergens bind to antibodies, not histamines. Option B. Antibodies bind to mast cells via their constant region, the tail. The variable region, the arms, must remain free to catch the allergen. Option D. This statement is factually incorrect. Histamine is an inflammatory mediator, not a chemical barrier. Furthermore, it is released after the Ig has already been triggered. During a hay fever response, the release of histamine causes localized vasodilation, leading to the swelling and redness typical of allergies. While complement proteins are specialized to neutralize cellular pathogens like bacteria and viruses, they remain inactive against non-living allergens like pollen. Meanwhile, macrophages function as the body's nonspecific cleanup crew, clearing foreign pollen particles through phagocytosis. In the lymphatic system, lymph nodes serve as critical hubs for the adaptive immune response. They are the primary site for clonal selection and expansion, where specific B cells are activated by antigens and rapidly multiply. While these nodes filter lymph, they do not produce antigens or allergens. Instead, they facilitate the production of plasma cells, which secrete antibodies to neutralize pathogens throughout the body. This question is related to primary versus secondary immune response. The correct answer is C. This graph perfectly illustrates the development of active immunity. Here is the breakdown of why graph C is the only scientifically accurate one. Initial exposure, day 0, there is a lag phase, roughly 10 days, while the body identifies the antigen and begins clonal selection. Primary response. A moderate amount of antibodies is produced, which then naturally declines over time. Second exposure, day 40. Because of memory B cells created during the first exposure, the response is much faster and significantly stronger. Secondary response. The antibody concentration shoots up rapidly and stays higher for a longer period compared to the first time. Why A, B, and D are wrong. A shows no decline after the first exposure. B starts with antibodies already present, which would be passive immunity, not active. D shows no lag phase and suggests an immediate, massive response on the very first day. Swine flu is a respiratory disease caused by a type A influenza virus that mostly infects pigs and on rare occasions humans. The World Health Organization, WHO, is monitoring both animal and human cases of swine flu around the world. In 2023, there were several reported human cases of swine flu. These cases have not spread easily from person to person. A case study is an ideal approach for the World Health Organization to track zoonotic diseases like swine flu because, real-world application, it provides a detailed analysis of actual situations and authentic data, which offers critical, evidence-based insights for scientists and global health organizations. Predictive analysis. By examining the specific causes and consequences of both historical and contemporary outbreaks, health bodies can develop timely, actionable recommendations to better manage or prevent future health crises. Key concepts to remember. In public health, these points relate to two major areas. Empirical evidence, using real-world data, ensures that policies are based on what is actually happening on the ground, rather than just theoretical models. Epidemiological planning, analyzing consequences, helps in resource allocation, like where to send vaccines, during future emergencies. MHC Class I markers serve a dual role in the immune response to the swine flu virus. Self-recognition. They facilitate the distinction between an individual's self-antigens and foreign non-self antigens, preventing autoimmune attacks. Antigen presentation. They allow virally infected cells to present swine flu-specific antigen fragments on their surface. Immune activation. This presentation signals for the activation and targeted destruction of the compromised cell by cytotoxic T cells or natural killer cells. Dendritic cells play important roles in the initiation of the immune response in many animal species, including humans and pigs. Dendritic cells have both MHC Class I and MHC Class II markers, as shown in the diagram below. Dendritic cells can be split into types based on their structure and function. The table below shows relative amounts of MHC Class II markers and interferon produced by two different dendritic cell types. Consider the information from the table above. Describe the different roles played by dendritic cell A and dendritic cell B in an immune response. Dendritic cell A, the antigen presenter utilizing its high density of MHC Class II markers, this cell primarily functions as a professional antigen-presenting cell, APC. It engulfs the swine flu pathogen via phagocytosis and presents foreign antigen fragments to helper T cells. This interaction is critical for initiating the adaptive immune response, eventually leading to the pathogen's systemic clearance. Dendritic cell B, the viral sentinel characterized by high interferon production and low MHC2 expression, this cell acts as a vital signaling hub. Its primary role is to secrete interferons that alert neighboring uninfected cells, stimulating them to produce antiviral defenses before infection occurs. Additionally, these chemical signals recruit other innate immune cells to the site, effectively slowing viral replication and accelerating the recovery process. This topic is related to responding to antigens and acquiring immunity. Mast cells play the key role in hypersensitivity. They release histamine to cause inflammation and vasodilation. B cells are involved into clonal selection. They multiply in lymph nodes to produce antibody-secreting plasma cells. MHC Class I is expressed in all nucleated cells and presents viral antigens to activate cytotoxic T cells. Dendritic cell A expresses high MHC Class II and acts as an APC to initiate the adaptive response via T cells. Dendritic cell A expresses high interferon and signals neighboring cells to create an antiviral state. If you need my help, please add me on WeChat or WhatsApp and send me an email. Thanks for your attention. Please note Dr. Liu's VCE Physics channel, Dr. Liu's VCE Chemistry channel, and Dr. Liu's VCE Biology channel.